Hello. Okay, so a pleasant good day to everyone. And in this video, we'll be looking at the immune and lymphatic systems. It is important to note that with the immune and lymphatic systems, certain themes should be recognized and appreciated. One is that every person has a set of proteins which defines themselves or the self, and these proteins are unique to that individual. With the exception, of course, of identical siblings, self-antigens are non-self, that is alien or foreign to every other person. So that sets of protein or the set of proteins which identify you as an individual, it's unique to yourself. The immune and lymphatic systems distinguish between self and non-self. Lymphatic structures filter the lymph fluid collected from the intercellular or the interstitial space, and in cells of the lymphatic system, they neutralize entrapped particulate matter and microbes. Finally, in terms of this chapter, and something else to appreciate is the immune system is a cellular defense system or mechanism that populates the lymphatic system with cells, and it is tightly integrated with it. All right, let's go forward with the presentation and see how this relates to it. When we're thinking about the cells of the immune system, what comes to mind is a barrier. If you look at this image here, you see the walls forming a blockade. So therefore, it renders penetration or the ability to get into this particular setup very difficult. So the city walls protect the occupants from external threats, very much like our physical and chemical barriers protect our body cells from pathogens. So I have a question out there. First question, what body system physically blocks pathogens from accessing our internal environment? Which system is that? Hmm. Correct is right. Very good. That would be the skin, which of course is part of the integumentary system, right? In terms of protecting the body itself. All right, so the integumentary system. Physical barriers are the skin and mucous membranes with hairs, mucus, and cilia present within them. The chemical barriers are saliva and stomach acid, and the pathogens, and the reason why they're there is because pathogens frequently gain access through the eyes, nose, GI tract, or the urinary tract. So therefore, we do have systems in place for protection. The immune system is the body's cellular defense system, whereas the lymphatic, this is a branching network of vessels and organs which house the immune system. So the protection, the immune system key players being the leukocytes or the white blood cells, whereas the lymphatic system is the place where they are housed. So what are the functions, you might ask, of the immune system? Well, one, defend against pathogens or the external non-self threats. Two, identify and destroy any abnormal cells arising from within, example, cancer cells. Have to remember cancer cells are what I like to call good cells gone bad. So they are your natural cells, but something has gone wrong, particularly as it relates to the replicative mechanism. Research has found that the retinoblastoma gene associated with the replication process, usually it is mutated in cancer cells. So the thing is, you don't want these bad cells around. It is up to the immune system to try to destroy them. Now, destroy, damage, or aid cells and free their components for reuse. That's what the immune system does. So it's a breakdown mechanism which facilitates the recycling of components of those things which are broken down. Let's talk a bit about the lymphatic now. The lymphatic system, it houses and supports the immune cells. It excretes and returns some interstitial fluid to the blood, and it absorbs fats and fat-soluble vitamins from the intestines. Very interesting. In terms of those fats, they are too large to pass through the capillaries. So therefore, the lymphatic facilitates that fact in terms of letting them pass through the lymphatic system, because the lymphatic system is a little bigger than the capillary system. So what are the cells associated with the immune system? Well, we have three classes. We have the granulocytes, lymphocytes, and the monocytes. The granulocytes, these ingest and digest foreign material, a process known as phagocytosis. And the lymphocytes, they consist of T, B, and natural killer cells. The monocytes, they develop intermacrophages and dendritic cells. It is important to note that the granulocytes, they make up about 65% of the blood leukocytes in healthy people. They are primarily inflammatory, and of course, inflammation is an immune activity when it attacks and neutralizes or destroys alien substances. Now, most of these lymphocytes are found in lymphoid organs and other tissues, which we will get on to in just a little bit. They complete their maturation in various lymphoid tissues. Monocytes develop into macrophages or dendritic cells, which are concentrated in lymphoid organs and body regions in direct contact with the environment, the epidermis, and the lining of the respiratory and GI tract, for example. Both of these are involved in antigen presentation. And when we look at these T lymphocytes and B lymphocytes, they point to where they are matured. T lymphocytes relating to the thyroid, B the bone marrow, where maturity of these cells occur. Let's go into a little more detail of these things. So the red bone marrow, we have the hematopoietic stem cells. They produce immature lymphocytes. So the immature lymphocytes are produced there. Two, some immature lymphocytes differentiate into natural killer cells, others into B cells. Right? So differentiation occurs in the thymus. Some immature um, lymphocytes migrate to thymus and become T cells. I think I might have misspoken in the last slide, said thyroid as opposed to thymus. Very easy to mix up. So I just hope you don't make that mix up, but appreciate it's the thymus where maturity occurs and not the thyroid gland. So the thymus, some immature lymphocytes migrate to thymus and become T cells. And the lymphoid organs themselves, the lymphocytes congregate in them, and such as the lymph nodes, and circulates through the blood and lymph. So this is just showing an overview of the lymphocytes themselves. So I have a question. Do natural killer cells differentiate in the thymus or in the bone marrow? Ah, very good. The answer, of course, is the bone marrow. Let's look at another type, the monocytes. Now, monocytes can differentiate into macrophages or dendritic cells. They do have that ability. Let me ask this question. Which monocyte descendant has a large number of thin processes? Well, if we were to look at these diagrams, these thin processes are hallmarks of dendritic cells. So if you answer dendritic cells, you'll be quite correct. So we looked at the particular cells. Let's look at the network associated with them. Now, the lymphatic network consists of capillaries, vessels, ducts, and nodes. The capillaries picks up the fluid from the tissues, whereas the vessels are formed by the merging of these said capillaries. The ducts carry fluid, to the large veins near the heart, whereas the nodes are small lymphoid organs that house many lymphocytes. And of course, they filter the lymphatic fluid to trap particles, bacteria, and other pathogens and foreign antigens. Very interesting to note. When you do go to a physician and he palpates your lymph nodes by feeling on your neck and the other areas where they're associated, what is actually he or she, what they're doing is actually checking to see if there are any trapped particles there, which is usually a sign of some pre-existing illness or current illness occurring within the body. So that's why the doctor palpates those lymph node sites to see if there are any infection which has occurred there, or if there are any trapped particles which are indicative of a previous infection or an existing illness. Now, in terms of this, do remember that the main lymphatic duct is the thoracic. If there's one takeaway from this, always remember, when in doubt, the thoracic duct. And this main thoracic duct, it drains lymph fluid from most of the body. It travels up the left side of the vertebral column and it empties into the left subclavian vein. Let's have a look at that diagrammatically. So when we look at the lymphatic system, here we are seeing in terms of the normal cardiovascular system, with the circulation from the heart to the arteries. And we have this parallel system, this lymphatic system, which occurs in which we see then the movement of lymph fluid through the capillaries and the various ducts, and of course, ultimately filtration occurring via the lymph nodes themselves. Now, the vessels of the lymphatic, they pick up excess tissue fluid and return it to the venous circulation. The tissue fluids enter the lymphatic capillaries through these small gaps between the cells and the walls. That is very important there. So I have a question for you. Which type of lymphoid vessels are physically connected to the cardiovascular vessels? Hmm. And that would be, very good, the lymphatic ducts. Right, very importantly, these are the ones which connect into the cardiovascular system, as shown here. All right, so now let's have a look in a little more detail in terms of the lymphoid vessels. So the lymphoid vessels, they connect all of the lymph nodes, and you're seeing all through the body the lymph nodes shown here, the axillary lymph nodes, and the vessels connecting them. These, of course, are the ones which are usually palpated by your physician in terms of the cervical and some mandibular in particular. These are the ones which are palpated usually. The thoracic and right lymphatic ducts, they drain into the subclavian veins, and the body regions are drained by the two lymphatic ducts. All right, so as shown here, in the ducts in this area are drained by the right lymphatic, whereas everywhere else they are drained by the thoracic duct. So I have a question for you. How are lymph nodes in the armpit region named? Our lymph nodes in the armpit region named, and they are named as axillary lymph nodes. Very good, axillary lymph nodes, right, in the armpit region as shown here. All right, spoke about the circulation. Now let's have a look at the lymph node itself. Now, the components of a lymph node, and here is shown, it has a cortex and a medulla, very similar to what is found in the kidneys, which we will look at in due course. All right, so the cortex and the medulla, here you have the afferent lymph vessel bringing the fluid to the lymph node, and of course the efferent taking it away. Here you have the medullary cord and the medullary sinus coming off of the cord itself. Now, I have a question. Are the germinal centers found in the cortex or the medulla? Well, the germinal center, if you notice it here, this is in the part which is shaped like a C. C is for cortex, so that answer will be cortex. Now, looked at the lymph node, let's talk now about the tissues associated or found in the spleen itself. So there are two broad categories, the red and white pulp. The red are broad venous sinuses and cords of splenic tissue, namely red blood cells, lymphocytes, and macrophages. The white are the nodules of lymphocytes and macrophages that cuff splenic arteries. Let's have a look at these diagrammatically. So this is showing the spleen here, and the thymus and spleen, of course, are located in the torso. The spleen is composed of both red and white pulp, and the lymphocytes make up most of the thymic tissue. They proliferate in the outer cortex and they migrate into the medulla. So very important to take note of those things. So I have a question related to the spleen. Are sinuses found in the red or the white pulp? Where are the sinuses found? Is it in the red or the white pulp? And if we are to look at the diagram here, we notice the sinus will is of course found in the red pulp and not the white pulp, which is shown centrally here. Very good. Let's go on. Now, malt, another type of tissue is malt so-called because it stands for mucosal associated lymphoid tissue. Now, the malt tissues, they guard common pathogen entry points. So these are very important. Notice the entry points are being guarded as related, shown here. You have the palatine tonsil, of course, for the oral cavity protection. Now, name the lymphoid tissue clusters found in the small intestine. So if it does get past here, get down into the intestine, which are found, what are the malt ones located there? Well, as shown in the diagram, these will be the Peyer's patches. Peyer's patches, because this is go through the intestine. Here you have, of course, the appendix, which is located in the lower region. But the Peyer's patches or Peyer's, these are the lymphoid tissue clusters which are found in the small intestine. Okay, so now let's talk about getting on to the immune part. Let's talk about antigens. Now, what exactly as it relates to your immune system, what is an antigen? Now, what they are is a molecule that's capable of stimulating a defensive response from the immune system. Such examples, glycoproteins, and these are responsible for ABO red cell antigen system. Note that the word antigen comes from the word antibody generating. That's why take the first part, antibody generating. So the antigens, they generate antibodies. And the antibodies themselves, these are specialized proteins that attach to and help immune cells destroy the antigens. So that is very important to take note of. So let's talk about some sources of non-self antigens. Two sources could come both from outside or inside. Those that come from the outside, it could come in via pathogens, be it viral or non-viral pathogens, and also from transplants. And within the body itself, these can come from cancer cells. And as we mentioned, cancer cells, these are cells that are gone bad. Very important to know that with viruses, they cannot replicate, of course, outside of a host cell. They have no membranes or organelles associated with replications. In essence, what they do is they hijack a host cell's metabolic processes. So viruses have to get into the cell. It's the job of the immune system to prevent them from doing just that. Or if they do enter the cell, to limit their spread. Non-viral pathogens, these are organized as cells and can live independently of other cells. They can cause damage by secreting toxins and, of course, depleting nutrients, which should rightfully go to your own cells. These breakdown products, when they are released, could incite inflammation within the body walls. Let's have a look now at this concept of non-self. Now, non-self antigens, these are, and again, do remember, antibody generators. They come from two sources: outside, such as pathogens, as we mentioned, and inside, such as the antigens of cancer cells. The pathogens could be viral, bacterial, fungi, amoeba, or worms. And in terms of the treatment for viral, use antiviral compounds or, note well, not antibiotics. Antibiotics are very specific for bacterial pathogens. Fungi and amoeba and worms, of course, they are treated with specialized drugs. Now, each pathogen class requires a different treatment. Antiviral compounds can shorten viral illnesses by slowing the viral replication process. Antibiotics interfere with bacterial metabolism but are ineffective against viruses. So it's very important to take note of that where that is concerned. Parasites themselves, if they do get it with anybody, they require treatment using specialized drugs. All right, so now let's talk about the MHC, or the major histocompatibility complex. And these are specialized glycoproteins found on the cell membranes of every nucleated cell. So these are special identifiers of cells. So different cells have a different combination of these glycoproteins. They are unique to individuals and they bind other self and non-self antigens. Do take note that incompatibility with the MHC antigens, these are responsible for most tissue and organ rejections after transplant. Let's have a look at these. So in a healthy cell, here you have this receptor here, MHC1, major histocompatibility 1, and these are located on the surface here. So it's shown here, the MHC1. I'm normal, healthy self, leave me alone. That is what it seems to be saying. Now the MHC glycoproteins, they display antigens on the cell surface that convey messages. These cells express MHC1 glycoproteins bound to normal cell proteins. Now, cancerous or virus-infected cell express, they do express MHC1 glycoproteins bound to abnormal cell proteins. Some immune cells, they express the MHC2, type 2 glycoproteins linked to proteins from invading pathogens. So on that, let me ask another question. Which type of antigen is linked to MHC2? Glycoproteins, one produced by one's own cells or one produced by a foreign cell. And as we just mentioned, those are the ones which are produced by foreign cells. So let's have a look at that. So the MHC glycoprotein as shown here, this is in an infected or cancerous cell. Hey, I'm dangerous, kill me. So non-self antigen, it is showing that message and inviting then other cells to come in to destroy it. Here's another one. Fragment of the captured alien antigen is displayed and it's saying find this antigen and destroy it. This is the task of antigen presenting immune cells. This is what they do. Do remember that MHC1 glycoproteins, they display internally produced antigens, whereas the MHC2 type, they display antigens from external sources. So these are one display internally produced antigens, are the ones for external sources. These are done with MHC2 glycoproteins. So we've seen the display, how they actually functions. Let's talk about these defenses, the defense lines which occur within the body. So your immune system, you have your normal body cells. So these are the pathogens you want to get in. What protection do you have? Well, first of all, physical barriers. These are the first line of defense against pathogens, right? And these are shown here. Anatomical and chemical include the skin mucous membranes and tears, within the eyes, preventing invasion through that area. Then you have the immune system. You have innate and adaptive. The innate immunity consists of phagocytes complement natural killer cells. The adaptive consists of T and B cells. So question, are T and B cells part of the adaptive or innate? And just by looking here, you could tell they are part of the adaptive immunity. Very good. Now let's talk about the complement system. Now, numerous complement proteins interact to form a membrane attack complex. Complement proteins coat the pathogens to render them easy prey for macrophages. So here we have a pathogen and a membrane attack complex, right? So here is shown that membrane attack complex, and after which time, because of this interaction, you do have cell lysis. So the membrane attack complex, here it is shown, right? And it targets it for destruction. You also have opsonization, where the individual component proteins which have been targeted or placed on the pathogen targeted for destruction by a macrophage. I have a question. How does the membrane attack complex, how does it kill it, the membrane attack complex? And the way it actually facilitates it is that it forms a pore that permits water entry, leading to cell lysis. So the destruction is targeted for destruction because the membrane attack complex causes water entry into the cell, leading to, in this case, the pathogen, leading to its destruction. All right, let's talk about how the cells of the immune system. Yes, as shown here, the macrophages, they identify the pathogen and they destroy it. Let's speak to how that cell activation occurs. Well, first of all, the pathogen binds to the B cell receptor. Here is shown the B cell receptor binds. Then this causes a primary response. So upon initial exposure, the activated B cell produces many clones, which differentiate into antibody secreting plasma cells. A few clones do not differentiate, but they remain as memory B cells. So some of them, here they are, but then you do have the cloning which occurs. The secondary response. So there's a primary response. The secondary is should it encounter these and should it encounter these antigens again in future, it would be able to launch or recognize or give off a response very quickly. In other words, this is one way in which the cell, if it does encounter the same antigen, at a later date is able to launch a response very quickly in terms of production of antibodies. So I have a question. What term or what is the term that describes the rapid proliferation of the lymphocytes? And that answer, of course, will be clonal expansion. And when you have more clones, that, of course, will mean that you have a lot more antibodies being produced. These antibodies will then go on and destroy the pathogen. So these antibodies, what do they look like? They look very much like a modified Y as shown here, and they have different areas associated with it. Now, each of the antibody monomers consists of two heavy chains. These are the heavy chains here, 1 and 2 light chains, 1 and 2. Parts of the light and heavy chains, they combine to form the variable region. So here it is, you have this variable region here, and the constant region. Variable region shown here, very important, and then the constant region as shown here. So collectively, the variable and constant forms the antibody. So I have a question. Where's the complement binding site? In the FC, in terms of the constant region or the variable region? And of course, if we look here, we'd see complement binding site is located in the constant region. Very good. All right. We looked at the action of the antibodies. Let's go into a little more detail here. So here's a macrophage. Now, antibodies agglutinate, or they gather together. They agglutinate pathogen cells, rendering them more susceptible to phagocytosis. The antibodies activate, of course, the complement pathway. So this is shown here, for instance. Here you have the antigen agglutination occurring, and now it's targeting occurs from the macrophage for its destruction. Of all of these pathogens, right? Because of the agglutination or aggregation of the antibodies leads to their subsequent destruction by the macrophage. This question, if you were to look at this diagram here, what two classes of antibodies do you see here? Well, you see the two classes are the IgG and the IgM. So in terms of the antibody actions, here you have the complement binding site. This is just showing the actions of the complement in terms of formation of membrane attack complex, enhanced inflammation, and opsonization and enhanced phagocytosis. All of these things are hallmarks of the complement system. And do remember that the antigen-antibody complex leads to pathogen destruction by phagocytosis or cell lysis. Receptors, the receptors on the cytotoxic T cells bind only to MHC type 1 proteins, which are present on all cells except, of course, red blood cells. All right, so now we're looking, we mentioned those previous ones. Let's look at the cytotoxic T cells. Now, a dendritic cell ingests an abnormal cell fragment, as shown here, degrades it into smaller antigen molecules, and combines the antigen with an MHC1 type 1 receptor, as shown here. So it degrades it, and it has it now, presents it as a receptor. The MHC1 antigen complex moves to the membrane, right? So here it is, where it interacts with a cytotoxic T cell with a matching receptor, right? Cytotoxic T cell matches with the receptor. The activated cytotoxic T cells divides to produce many clones. This is known as clonal selection. And these clones include some memory cells. The activated cytotoxic T cells bind the abnormal antigen displayed by the infected cells. So in other words, then, after presentation, the cytotoxic T cell, it clones itself and now is able to go out looking specifically for an MHC type 1 presenting this antigen so that it could target it for destruction. And in terms of the release substrate that induce apoptosis of the infected cell, right? So here you have the infected cell, it does have the viruses, but the cells themselves, they present fragments of the virus in terms of on the MHC type 1 receptor. These cloned cytotoxic T cells then recognizing that particular antigen or unique protein structure, which is part of the virus. Now it binds to it and it induces apoptosis to the cell. Apoptosis programs cell death, so it leads then to the destruction of the cell. In so doing, the cell is destroyed, but also, more importantly, all the viruses which are present within it. This limits then the spread of the virus itself. A very handy mechanism. So that's the cytotoxic T cells. Now, in terms of the helper T cells, the steps involved in helper T cell activation is that any antigen presenting cell can activate a helper T cell if it expresses the relevant antigen attached to an MHC type 2 receptor in its outer membrane. The helper T cells induce an immune response by secreting cytokines and physically interacting with the B cells. So as shown here, one antigen presenting cell, it internalizes a pathogen, processes it into smaller antigenic molecules and combines the antigen with an MHC2 type receptor. The antigen complex moves to the membrane, as shown here, where it interacts with a helper T cell with a matching receptor. This T cell divides to produce many clones. This activated helper T cell secretes cytokines, so specifically which activate cytotoxic T cells and promote innate immune activity such as inflammation and phagocytosis. After this is done, the helper T cells bind to and stimulate B cells displaying the same process antigen. And this is how the process occurs, leading to the destruction of these pathogens. I have a question. Do helper T cells interact with antigen presenting cells, MHC class 1 or MHC type 2? And of course, as we mentioned, the helper T cells interact with the MHC class 2. Right? And here it is, pathogen binding, right, to the receptor, the B cell receptor, as shown here. Right. So lymphocyte production and action, the lymphoid, it just shows the differentiation in terms of the summary of the production, maturation, activation, and action of B and T lymphocytes and the natural killer cells. Note that the helper T cells stimulate the activity of all other lymphocytes, and in which are shown here. So the lymphoid stem cells, you have two types. You have the immature T and the B cells. They migrate, of course, to the thymus for maturation and the bone marrow, respectively, where maturation occurs. The T cells, as shown here, they are associated with MHC class 1 type receptors, whereas when the B cells, they're activated by stimulation from the T cells, and this, of course, then leads to production of antibodies in terms of their actions. So what type of lymphocytes attack cells with low MHC1 levels? And that answer will be, of course, your natural killer cells. So the last part we want to concentrate on is the active versus the passive immunity. Now, the immunity refers to the ability to resist infection by a particular pathogen, Active is immunity provided by antibodies and memory cells, whereas the passive is immunity provided by transferring pre-made antibodies into a non-immune recipient. So the natural, these example are antibodies in breast milk, whereas the artificial ones, these are the ones associated with vaccines. So as shown here in terms of the immunity, yes, active and passive. Was an antigen administered? Yes. Active, this is a type vaccination with antigen. If not, exposure to antigen in the environment. So this just shows how these acquired immunities, how they actually exist. Active immunity, yes. Passive immunity, no. In terms of it relates to the antibody and memory cells, which were self-produced. All right, and this is just again showing the response to bacterial infection, shown in A, whereas in B is showing a response to viral infection. All right, so in terms of these diseases, the autoimmune diseases, conditions in which the body attacks self, antigens, and allergies, of course, these are exaggerated immune reactions to certain substances. So these are examples when the immune response is not always helpful. All right, so we'd stop there in terms of the presentation. And I do hope you enjoyed it, and stay tuned for further ones as we go along this path. Okay, have a good one. Take care. Bye bye.